I'd like to thank the organizing committee for giving me this chance to talk to you all today. And today, I'm going to discuss what's being done to identify the genetic mutations that cause GLA, GSD, and KLA. But before I go into the work that's being done on these rare diseases, I just want to briefly go over some basics in genetics. And we'll start by going over the hierarchy of the human body. So the human body consists of multiple different organ systems. There's the digestive system, the cardiovascular system, the urinary system, and as well as several others. These organ systems are made up of organs. Organs are made up of tissues, and tissues are made up of cells. And when we look in cells, we can see that they contain this large molecule called DNA, and that's going to be the focus of today's discussion. So DNA is a molecule that contains all the instructions for the development and function of an organism. It's this molecule that's composed of two chains that form a double helix, and each strand of this molecule is made up of the building blocks G, A, T, and C. So A on one strand pairs with T on the other. I'm going to see if I can use this other pointer. Well, so A on one strand pairs with T on the other, whereas G on one strand will pair with C on the other strand. This molecule is packaged into a thread-like structure called a chromosome, and most of the cells in our body have 46 chromosomes. So DNA is important because it codes for proteins. So there are specific regions of DNA that are transcribed into this molecule called mRNA. mRNA is then translated into protein. And proteins are the molecules in your cells that perform pretty much all of the functions within a cell. So proteins, like I said, they perform an array of functions. Some proteins serve structural roles. So some proteins in your cell form the skeleton of the cell. Some proteins catalyze chemical reactions, and those types of proteins are called enzymes. Other proteins relay information between cells, and other proteins control the activity of a cell. So some proteins will tell a cell to grow in size, some proteins will tell a cell to divide, and other proteins will cause a cell to migrate and move around. And genetic mutations are bad, in part because they result in the formation of defective proteins. So what we're looking at here is a genetic mutation, shown in red, where a GC base pair was changed to an AT base pair. So this genetic mutation that's present in the DNA is transcribed into mRNA, so the mutation is present in the mRNA, and this mutant mRNA is then translated into mutant protein. And this mutation causes this protein to no longer have its normal function. So because this protein isn't functioning properly, the cell doesn't function properly. These abnormal cells then form abnormal tissue. The abnormal tissue then affects the biology of the organ, which affects the biology of the organ system and the organism. And that's how we can see disease. So I just want to briefly go over two different types of mutations that occur. So we'll discuss germline mutations and somatic mutations. So germline mutations are mutations that are present in the DNA of either the sperm or the egg. In this case, the mutation is present in the sperm. The sperm fertilizes the egg, and an embryo develops. And because the mutation was present in the sperm, every single cell in this embryo has that genetic mutation. This embryo then grows up to be an adult, And because this mutation is present in every cell in this adult, it's also present in his sperm. And he's able to pass this mutation on to his offspring. A somatic mutation is different. The somatic mutation is a mutation that is not present in the sperm or the egg. So in this instance, a mutation randomly occurred during embryonic development. And you see the presence of this red affected tissue in this embryo. This embryo then grew up to be an adult male, and you can see that the mutation is restricted to this affected area of tissue. The mutation is not present in every cell in the body, and it's not present in the sperm, so this mutation is not going to be passed down to his offspring. So we think of germline mutations as mutations that cause inherited diseases, whereas sporadic mutations or somatic mutations can cause sporadically appearing diseases that do not display a pattern of family inheritance. And there's evidence coming out now that some sporadic lymphatic anomalies are caused by somatic mutations, not the germline mutations. And when sporadic lymphatic anomalies occur, we think that the mutation occurs in the lymphatic endothelial cells, so the cells which make up lymphatic vessels. So in this case, what we're looking at on the left is just a schematic of some lymphatic endothelial cells, and a mutation randomly occurred in a lymphatic endothelial cell. Those mutant lymphatic endothelial cells are shown in red, and you can see that this mutation is passed on to the daughter cells, and these mutant lymphatic endothelial cells go on to form abnormal vessels, whereas the normal lymphatic endothelial cells form normal lymphatic vessels. And normal lymphatic vessels function as the body's drains. So blood vessels lose fluid, and this fluid is picked up and drained by lymphatic vessels. Lymphatic vessels also drain other substances like fat, and they also transport and move cells. If the lymphatic system doesn't develop properly because there's a mutation, then that means that the drains aren't developing properly. And if the drains aren't functioning properly, that can cause fluid to accumulate in areas. This can cause swelling like lymphedema. It could also cause chylocystis and chylothorax. So, what's going on with these sporadic occurring lymphatic anomalies like GLA, KLA, and GSD? We think that there's a genetic mutation, and that this mutation then causes the protein to not function properly in lymphatic endothelial cells. This abnormal protein causes the lymphatic endothelial cells to misbehave and not act like normal lymphatic endothelial cells. These abnormal lymphatic endothelial cells then form abnormal vessels. And because the vessels are abnormal, they're not going to be good drains. And that's what we think leads to this accumulation of fluid in tissues or leakage of fluid in tissues. So why are we interested in identifying the genetic cause of a disease? Well, if we know what gene is mutated, then we will know what protein is messed up. And if we know what protein is messed up, we could potentially use a drug to block the activity of that mutant protein. So by treating with this drug, we could potentially cause the mutant cells to act like normal cells. Alternatively, we might cause the mutant cells to undergo apoptosis or die. Or we could simply just arrest the growth of these mutant cells and arrest the progression of disease. So by knowing the genetic cause of the disease, that will point us towards a specific therapy or give us an idea as to what therapy we should use. Also, knowing the genetic cause of a disease helps move research forward. And if we know what genetic mutation causes the disease in people, we can engineer animals to have those mutations. And that then provides us with an animal model of the disease so we could better understand how the disease progresses, and we could also try to identify biomarkers and therapies for the disease in mice or other animals before we try these things in people. So this is just an example, actually, some work that we've done in the lab, where what we're looking at are lymphatics in the skin of mice, and you can see a normal mouse, this network of lymphatic vessels, and a mouse that has a genetic mutation in lymphatics. These mice develop this very irregular network of lymphatics that don't function properly. So we're using these mice to develop a better understanding of how the disease progresses, and we're also testing therapies. So how are researchers going about identifying the mutations that cause GLA, GSD, and KLA? First, we think that these diseases are caused by somatic mutations. So that's our hypothesis. It's not a germline mutation where you could detect the mutation in just any cell in the body. We think that we have to focus in on affected tissue. In this diagram, the affected tissue is shown in red. And if you zoom in on the affected tissue, you can see that it consists of normal lymphatic endothelial cells, which are shown in green, red mutant lymphatic endothelial cells, and then there's just a bunch of other normal cells within this affected tissue. All of these cells that are present there, all of the normal cells, make it difficult to identify the mutation in just the affected tissue. So if we were to just sequence this affected tissue, we might miss the mutation that causes the disease because all the normal cells might mask the mutation or affect our ability to detect it. So what investigators are doing now is isolating lymphatic endothelial cells from the affected tissue. So we're enriching for the cells that we're interested in. When we do this, we not only get the mutant endothelial cells, but we also bring along normal endothelial cells as well. But we enrich for the cells that we're interested in analyzing. These cells are then grown in a dish, in a lab, and then ultimately DNA is isolated from these cells and sequenced to identify the mutation that causes mutation in the patient. And what we're looking for, too, are mutations that are present in the affected tissue, but not in the unaffected tissue. So once the DNA is isolated, there's different types of sequencing that's performed to identify the genetic mutation. You might hear of exome sequencing or whole exome sequencing. And this is a technique to sequence all the protein coding genes in the genome. So all the regions of DNA within the genome that code for proteins or the workers of the cell. There's also whole genome sequencing, which is a technique to sequence just the entire genome, all the base pairs of DNA. So both are used. And I just want to briefly go over a success story. This is a paper that was published earlier this year, and it's on one patient that was diagnosed with GLA, and it was a sort of a GLA, KLA type patient. And what we're looking at here is affected tissue from the patient. What the investigator did was isolated lymphatic endothelial cells from this affected tissue, cultured these cells on a dish, and then sequenced this DNA isolated from the cells. And by doing that, she identified an activating mutation in a gene called NRAS. And now what this is allowing us to do is it's not only giving ideas or pointing towards specific therapy, but it's also led to the development of an animal model of the disease. And what this investigator has done is created a zebrafish model. And she's using this model to test different therapies and to better understand the progression of this disease, or to understand how NRAS drives lymphatic abnormalities. So I just want to summarize by saying that genetic mutations are, a genetic mutation is an irreversible change in an organism's DNA, and we hypothesize, or we believe, that lymphatic anomalies are caused by somatic genetic mutations. And the exome sequencing is being performed to identify these disease-causing mutations. And we think that this information will guide the search for therapies and will also lead to the development of new research tools. So thank you for your attention, and I'm happy to answer your questions during the question session.